Assuming that we've already uploaded our qPCR data into the application, it is now time to define the experimental conditions of the analysis. These conditions can be defined in two different but compatible ways, by loading them from an already predefined text file or creating them from scratch by editing the design panel. First option assumes that we have a tabular text file for editing this format. As we can see, there is a mandatory column called the sample name, including the sample name defined in the input files, and a variable optional number of columns for each experimental factor that we want to include in the analysis, in this case tissue and treatment. As an additional step, plate is an optional and useful column when we have two samples with same names on different plates. To apply this design, we only need to select this file from Experiment Design Browser and ensure that every factor has been properly loaded. As we can see, our example consists of 3 brain tissues, amygdala, cerebellum, and a reference group, and each one has been replicated 3 times. In this case, the factor treatment is not really needed, and we can remove it by right-clicking and selecting the Delete Select Factor option. If we want to add a new factor, we can use the same menu and proceed to fill it in manually. There is also a paired factor option in cases where samples between biological groups are paired by another factor, such as time, and therefore we would like to perform differential expression analysis including this information in the statistical test. This factor is created in an equivalent way by assigning the same group or ID to those samples that are paired or have any kind of relation. If this information is defined, then StatMiner will automatically include it when a paired test is executed in the differential expression analysis. That is all. We can now save our design as a text file to use it in the future if needed. Before clicking the Load button to apply a design, we have the chance to exclude from our project any sample that we do not want to include in our analysis. To do that, just uncheck them from the left-hand side box of the design panel and they will be automatically discarded. So, this is our final experimental design. StatMiner will automatically create a new factor termed the master experimental factor as a result of combining condition factors such as tissue and dose in this case. This is a default selected experimental factor and will allow us to analyze the effects of all the associated conditions. Anyway, we can select any of those factors to define our analysis, but it is important to know that our results will be affected in different ways depending on the defining factor, and not only in terms of groups that we can compare at differential expression steps. Biological groups and distribution affect the imputation of missing values and best endogenous control selection, but also in general to all quality tests applied to the analysis as biological outlier detection, amplification flags, etc. To finish this tutorial, let us go back and propose a slightly complex scenario. Although these are the same files, it is a slightly different case. For instance, amygdala 1, 2, and 3 are not biological but technical replicates. It means that the corresponding wells on different plates contain the same sample. It is very important to tell StatMiner how to deal with such replication. For example, does our experiment consist of 3 tissues and 3 biological replicates by tissue? Or are we talking about 3 technical replicates by tissue? By default, StatMiner will treat the same sample name on different plates as technical replicates. According to that, we have to rename amygdala 1, 2, and 3 with the same name so that StatMiner can treat them as technical replicates. To do that, we will open the Rename Sample tool and give the same name to these replicates. If needed, we can be supported by the search tool. So let us proceed to rename the other replicates in the same manner. As suspected, this rename sample tool can also be used to rename confusing sample names. So now that we have given the same name to the technical replicates, what will happen is that StatMiner will run an analysis consisting of 3 different tissues with no biological replicates. A very similar case where the sample with the same name on different plates should be treated as biological replicates can be fixed by proceeding to rename each sample. Please watch our subsequent tutorials to get a deeper understanding of StatMiner.